Having a few camera issues, but what we're going to do today is define several of the terms on your sheet, enough to get you by. It'll be short. It's not the greatest because this stuff is kind of cool, but I haven't got the time to do it justice. So I'll run through, maybe give you a few examples. First thing, what we're going to do is define evolution. Of course, we're talking about biological evolution. Biological evolution. Because, of course, galaxies evolve, solar systems evolve. We're talking about biological evolution. There's two ways to define it. There's kind of a more modern definition and an older one. The modern one is called microevolution. The older, more traditional one is macro or macroevolution. So we'll do microevolution first. Microevolution. What that is, it's basically a change in gene frequencies over time. You look at genetics last semester. There's a gene out there in the human population that controls the development of your hairline. One form of the gene gives you a straight hairline. I'll use S. The other form of the gene gives you a bit of a dip down in the middle, kind of like the Eddie Munster thing going on, if you exaggerate it. That is called a widow's peak. I'll use a capital S to represent it. Now, if you look in the human population right now, and you sampled everybody, or at least a representative sample, and let's say you found in 2021, you find, there, so you can see it, just a date. You sample a population, you find of all the alleles in the human population, 90% are of the straight hairline variety. The remaining 10% are widow's peak. Then you come back after a generation or two, let's say 2071. 50 years is about 2 generations. And you do the same thing. But you find 85% of the alleles are the straight hairline, 15% are the widow's peak. That's a change in gene frequencies. That's what microevolution is. Short time interval, and you can demonstrate it in the lab. Humans aren't the best. We have long generation times. But with fruit flies, you can show change in gene frequencies. So it's basically short time interval. It doesn't take long to get a small change in gene frequencies. But the idea is, for the classic macroevolution, you accumulate these small changes over enough time and you could have a completely different species. That's the idea. Sorry, I'll do some splicing together. I'm having camera issues. I lost one, my other one's dead, and this one is actually the microscope cam we use in the lab. I just taped it to the faucet, so I'll have to do a little splicing. But anyway, and I know it's even harder to read the resolution's a little credit. But we did microevolution, change in gene frequencies over time. What we're going to look at next, the other definition for evolution, is macro or macroevolution. This one, you can kind of define it as a change in the species composition of the entire planet over time. Like, for example, if you went back in time, 100 million years ago, looked around on the planet, what would you see? Dinosaurs, things like that. You fast forward 100 million years, they're not there, and there's these other funny-looking mammal things around, and then dinosaurs are just kind of left as little birdies. That's macroevolution. They're basically the same thing. It's just a matter of degree. Microevolution, small genetic changes over a relatively short period of time. If you lengthen the amount of time, you get accumulated changes in the genetic makeup, which could eventually lead to different species or speciation, which we might get to in a bit. So that's it. Those are our definitions for micro and macroevolution. The time frame, how fast does evolution occur? There are different ideas, different theories, probably a combination of both. The old idea, or the old sort of theory, which is still valid, is called phyletic gradualism. That one kind of describes evolutionary changes occurring very slowly in small bits and pieces over a very long period of time that accumulate into much bigger changes. The other one is a punctuated equilibrium model. That one suggests that there's long periods in the evolution of a lineage where there are not many significant changes, and all of a sudden, what appears to be overnight, in a geological sense, all the organisms change and they have this new trait. So a long period of equilibrium with no significant changes that you can see from the fossil record and such, and then in a very short period of time, all the species, all the individuals, or that lineage has changed radically. It'd be like clams. You look at a bunch of clams in the fossil record. Some of them might be kind of smooth. And as you look kind of more frequent and more recent in time, you might start to see small bumps on the shell develop. And then later, there may be little spurs. And then later, these big spikes. The idea is a little bit of toughness in the shell and predator avoidance mechanisms would slowly be increased if they were favorable for the reproduction and survival of that group. That's phyletic gradualism. Punctuated equilibrium, you might see fossils of these basic clams. They stay the same, and they stay the same, and they stay the same. Then, in the next rock strata, they have these big elaborate spines with really no transitional individuals that we have records of anyway. That's punctuated equilibrium. Long periods of stasis or no change, and then a very rapid change where all the individuals acquire that new form over several generations. I should say the lineage acquires the new form, individuals don't. So that's kind of the time frame. And again, it can be both. You might have a single lineage that experiences phyletic gradualism, and then there may be a period where there's some punctuated equilibrium. So there's probably a mixture of the two. That's a general idea. Now, the last thing we'll look at, I know it's quick. That's our definition for evolution. What we have to look at next is how can we explain it? What is a mechanism that can explain how evolution could occur, either micro or macro? We'll look at several. We'll look at natural selection. That's a mechanism. That's the one Darwin proposed in about 1844. I might have that date wrong. We'll look at neutral or genetic drift. That's a second mechanism. And artificial selection. That's just kind of fun. That's a man-made one. We do it. Natural selection. To not take a lot of time with natural selection, I'm going to give you kind of quick 4 points. Darwin's thesis was a little longer, but kind of 4 main points. You can slow down or listen to the tape if you need to. I'm not going to write them on the board because I don't think you can read it with this camera anyway, especially with my penmanship. It's almost as bad as Christopher's. Anyway, you've got in natural selection, one of the ideas, and these are all observations. We have a theory, the bare bones of it anyway, before we knew much about genetics or before we rediscovered anything about genetics. But what it is if you look at any species out there, they tend to have a very high reproductive potential. Usually that reproductive potential is not realized. That is, individuals have a lot of offspring. Most of those offspring do not survive. And if they did, every species would be experiencing exponential growth, and the planet would be covered in 2 or 3 feet of dead and dying organisms, and that would be the end of that. So the idea is individual organisms have a very high reproductive potential, but they don't realize it because most of the offspring will die. Then, sort of number 2, those offspring, they all are genetically slightly different if we're looking at sexually reproducing species anyway. Because of independent assortment of the homologous chromosomes crossing over, and of course throw in a little bit of mutations, you have differences. If you have a bunch of siblings, you don't all look identical. You look similar, but you're all a little bit different, unless you're identical twins. So the idea is, all right, if you got all these offspring, most of them aren't going to survive. Which ones survive? And I think Darwin used the term struggle for existence, and the term survival of the fittest was introduced later. But what it basically means is the individuals that have a genetic makeup that allows them to reproduce most successfully in the current environment, those genes tend to be favored. Why? Because if you survive or reproduce a lot, you'll have more of those genes will be represented in the next generation. If you have a genetic makeup that isn't that adaptive, it doesn't improve your reproductive chances at all, then you're going to have fewer offspring. Your genes will be represented at a lower frequency as we go through the generations. So that's a general idea. In fact, there's 3 words you need to remember with natural selection. That's really all it is. It's differential reproductive success. When they talk about fitness, in an evolutionary perspective, fitness is really the number of offspring you produce. You can be physically fit, that doesn't matter. If you have no offspring, your fitness is 0. Your genes are not going to be in the next generation. That lineage is sort of terminated. So fitness is the offspring you produce. No offspring, no fitness. The more offspring you have that reach reproductive age themselves, that is your fitness. So kind of in a nutshell, I'm going to leave it at that. Lots of examples. There's a lot of cool stuff goes on in natural selection because it selects for some pretty bizarre traits. Transvestite scorpion flies, deer with these funky looking antlers. What are they good for? Not much, except attracting mates, so they improve your reproductive success. Then, trying to keep this short without leaving out too much. So that's natural selection. Now you will see, my sheet of terms and textbook, they'll talk about different types of natural selection. It's all the same, but there's directional selection, disruptive selection, and stabilizing selection. They're all natural selection, but they can affect the population in different ways, and they can affect the same species at different times. One species might undergo directional selection and stabilizing. Basically, in directional selection, we plot a population using kind of the standard bell curve. So the trait, so on the x-axis, y-axis is really the number of individuals. And then if you look at it, you can pick any trait. You can pick height in humans. An adult height of 4 feet, an adult height of 8 feet, an adult height of, what do you think the average adult height is of humans in Kentucky? 5 feet, I don't know, 10 inches if you combine males and females. I'm not sure exactly what it is, somewhere in there. So the average is right here. And as you'd expect, most people are around average. These tails of this curve, they're referred to as phenotypic extremes. That is, if you see an adult, walking down the hall and they're under 4 feet tall, I know it's rude, your mother tells you not to, but you're kind of going to look at it a little bit because it's unusual. You go, wow, that's a real short person. No judgment there. It's just unusual. If someone walks by, that's 8 feet tall, you look up, oh my gosh, that's a tall person. They stand out because they're unusual, because by definition, these phenotypic extremes, they don't represent that many people. Most of us are around average. Someone walks by and it's 5'10", give their height a second thought. In directional selection, what it means is natural selection, selective pressures, whatever they are, predation, disease, food acquisition, whatever. If you select against one phenotypic extreme, for example, let's say all the tall people, for some reason, didn't reproduce. What's going to happen to the average over time? This whole curve will shift the other direction. So it's moving in a direction. You'll change the average height. I mean, if you remove all the tall people and all the genes that favor tallness, over time, that population's average is going to be smaller. Mathematically, you get rid of the 8-footers, average height goes down. That's directional selection. It's selection against one of the two phenotypic extremes, and it's going to move the average. That's directional. Stabilizing. You can start off with the same population curve. In stabilizing selection, both phenotypic extremes. If you get rid of real short things and real tall things, whatever it is, humans, plants, selection gets both. What's that going to do to the average? It's going to keep it right where it is. It's just going to take this whole bell curve and kind of narrow it and keep it close to average. You typically see stabilizing selection in a species that's been living in a stable environment for a long period of time. Think the ocean, out in the deep ocean. The animals have been living there for a long time, let's say horseshoe crab, millions of years, tens of millions of years, and the environment doesn't change. So all the individuals are pretty well suited to that environment right now. Any changes in that genetic makeup from sort of the average are usually detrimental. That is stabilizing selection. So as you can see, something like horseshoe crabs now might be experiencing stabilizing selection, but then if the environment changes, water temperatures go up or something, that's going to favor one of the phenotypes. So you might have stabilizing selection. Then if the environment changes or there's a new mutation, you might get directional selection. The last one is disruptive selection. That one is harder to describe. For me, it's hard to explain. That's selection against the average, which means the individuals with the high fitness are at the extremes. The example I'll use is snails. There's an intertidal species of snail, crawls around between high and low tide, scraping algae off of rocks. This species lives in, little tropical volcanic islands out in the South Pacific. Now, those islands, they have rocky basaltic sort of rock outcrops, the volcanic rock. And then there's eroded areas where the sand is deposited in nice and white. So the environment is white, or it's, brown, dark brown, or black. The snails come in 3 color phases, genetically determined. There are white snails, there are dark brownish black snails, and there are pink snails. Now, these snails aren't very smart. They're gastropods. They crawl around randomly. Everything likes to eat them. Birds, if they can get them. Fish, if they can get them. One of their main protections is just cryptic coloration. They blend in. They're camouflage. So if you're a brown snail randomly crawling around, sometimes you're on those dark brown rocks. You might survive and you reproduce. If you're a white snail randomly crawling around, sometimes you'll be on the white sand. You blend in. You might survive to reproduce. If you're a pink snail, you're screwed because they can see on whatever you're on, and everything eats you, so your survival is low, and therefore if you're dead and eaten, you don't reproduce that much, so your fitness will be low. Disruptive selection is selection against the average phenotype. The ones with the highest fitness are the two phenotypic extremes. They'll go up in numbers of individuals. The average will actually be selected against, and it will go down. So that is three types of natural selection. Now, last thing or two, I'll just define them because I don't know how useful this is. Much better with examples. But you've got artificial selection. Artificial selection is kind of cool. As the name implies, it's artificial because humans do it. In artificial selection, humans pick the traits that are passed to the next generation. We've been doing this probably for tens of thousands of years before we knew anything about genetics or anything else. All our domestic animals and plants are made this way. Take German Shepherds, for example. Breed German Shepherds together, they all look like German Shepherds, but they're still a dog. They're still Canis familiaris. They're still that species. To give you an example, all the new dog breeds, there's new ones coming up all the time. You can set criteria for them. Let's say you wanted to make a teacup St. Bernard. All right, St. Bernards are 150 pounds as adults. Teacup, I don't know the Kennel Club rules, but that's something like 6 inches at the shoulder or something. It's those really tiny dogs you can put in your pocket kind of thing. How do you go from a St. Bernard at 150 pounds and get it down to something this big? Well, what you do, you use artificial selection. You get a bunch of St. Bernards. The bigger your starting population, the better. And you breed them together. And you'll notice the puppies, they're all a little different. You always have some runts in the litter, the smallest ones. Well, guess what you do? You keep the runts, you get rid of all the others. They refer to them as culls. I don't know, sell them to pet stores, whatever. You don't keep them. They're not in your breeding population anymore. You grow the runts up. You breed them together. You take their smallest puppy, keep it, get rid of the others. And you keep doing that. And what you're doing is you're selecting for any of the genes that make you smaller. Any largeness genes you're removing from your population. Over time, you'd be able to reduce the size of that, St. Bernard substantially. Now, could you get to teacup? Probably not. But you could probably get to a 40-pound St. Bernard pretty easy. Then there's other complex things you'd have to do. Once you got to your 40 pounds, you basically probably couldn't get any smaller because you only have the genetic variation to work with that was in your original population. You might not have any genes that would make it as small as a teacup. So then what you do, you take your somewhat smaller St. Bernards and you outbreed. You'd go get a Chihuahua or something, and you breed your Chihuahua with your smaller St. Bernards, and then you get pretty small puppies. But they wouldn't look like St. Bernards. They'd be Chihuards. I don't know what you'd call them. St. Chihuahuas. But they'd be smaller. Then you'd have to take all those puppies, pick the smallest ones that also look more like a St. Bernard, get rid of the ones that look more like Chihuahuas, and so forth. Keep going, keep going. And eventually you'd get something that looked a bit like a St. Bernard that was really tiny. That's artificial selection. Humans pick the genes. Not always good because we pick some pretty crazy things, like domestic turkeys that can't fly and are really, really stupid. But they develop really large pectoralis muscles, the breast meat. Last thing I'll mention quick, then I'm just going to wrap it up. Neutral selection or genetic drift. This is a special case. Neutral selection or genetic drift is a change in gene frequencies due to random chance. None are better than the other. It's just, if you have a small population, and that's a restriction for genetic drift, it only occurs in very small, isolated populations, which is important when you're looking at, conservation genetics and small threatened and endangered species or population sizes get really small. And basically what can happen, you can have an allele that just by random chance doesn't make it into the sperm and eggs that are used to make the next generation. It's gone. So, and I'm just going to leave it at that. Genetic drift or neutral selection is a change in gene frequencies really just caused by random chance. The gene you lose could be a good one. And that's a problem with small, isolated populations. They tend to lose genetic variation. And of course, in the long run, that's usually detrimental for the population. They're less, they can adapt to or adjust to change in the environment to a lesser degree than if they had a lot of genetic variation. All right, I'm going to see if I can actually download this and use it for anything. If I can, I'll post it to Blackboard, and that's about all I'll get. Just kind of look through the chapter a bit. I'll throw a couple questions on the next exam, and that'll have to do it. Take care.